Alright, so welcome to my YouTube channel. Today I'm going to be taking something very important for your exam. Structure and synthesis of DNA. Please, I will beg you to take this very serious. So I'm going to be looking at the structure of DNA, then the synthesis of DNA. Now, first, we should note that the DNA is one type of nucleic acid. We have two types of nucleic acid, DNA and RNA. And the building block of DNA, the building block of nucleic acid is called nucleotides. So nucleotides are the building blocks of nucleic acid, including DNA. And the nucleotides are composed of three things, which are nitrogenous bases, pentose sugar, and phosphate group. So these are the three components, these are the three compositions of the nucleotides. So it is when we link one nucleotide to another nucleotide to another nucleotide that we have our nucleic acid. So nucleotides are the building blocks of nucleic acid. Now if you look at the structure of DNA, the DNA is a double-stranded helix. The DNA is double-stranded, meaning it has two strands. And the two strands of DNA, one of the strands is called the template strand, and the other strand is called the complementary strand. It's called the complementary strand. Now the template strand, take note that the template strand always runs from 3' to 5', while the complementary strand always runs the opposite direction, 5' to 3'. So you can see that the two strands are anti-parallel because they are running in opposite direction. All right, now inside we have bases here. And there's what we call base pairing. In that, in base pairing, adenine we always pair with guanine with 3 hydrogen bonds, and adenine will always pair with thymine with 2 hydrogen bonds, and guanine will always pair with cytosine with 3 hydrogen bonds at Church of God. That was the nonsense I used when I'm doing this long ago. Alright, so A pairs with T with 2 hydrogen bonds, and G pairs with C with 3 hydrogen bonds. What that means is, on this template strand, if we have G here, what will be here? Will be C, and hydrogen bond, 3 hydrogen bond is something in between. If what we have here is A, what will be here is T, and 2 hydrogen bonds will be formed. What is here is T, what will be here? A, right? 2 hydrogen bonds. If here, what is here is C, what will be here? G. How many hydrogen bonds? 3. So you can see that what is holding the two strands of DNA together is hydrogen bonds between the bases. That's what is holding the bonds together, the two strands together. So basically, the structure of DNA, DNA is double helix. It's a double helix structure, having the template strand and the complementary strand. And the bases on both the template and complementary strands, they are joined together. The two strands are joined together by what type of bond? A hydrogen bond. You should also be able to tell them that the template strand runs from 3' to what? 5', while the complementary strand runs from 5' to 3'. Now, I'm going to be taking the synthesis of DNA now. DNA. One thing we should note is DNA synthesis is semi-conservative. Let me explain. You have this DNA strand. For synthesis to take place, the first thing is these two strands will unwind from each other. And the only thing that can cause these two strands to unwind from each other is the breaking of these hydrogen bonds, right? So the hydrogen bonds will break from each other and they unwind. But now look at this. This is our nucleus. And the DNA is found on the chromosome. Long strands. So where will this unwinding start from? That particular point on the long DNA strands where the unwinding starts from is called, let's say the unwinding is starting from here, is called the origin of replication. It's called origin of replication. So that particular point where the unwinding starts from is called origin of replication. Now, I'm going to talk more about that, but now let's look at this. From DNA to DNA to mRNA to protein. Now, from DNA to DNA, that's why using DNA to synthesize DNA is called replication. Then from DNA to mRNA is called transcription. Why from mRNA to protein is called translation. This DNA mRNA to protein is what we call the central dogma of molecular biology. So this is the central dogma of molecular biology. So if you ask for the central dogma of molecular biology, DNA to mRNA to protein, that's that. Now, so I've said that the two strands will unwind from each other. The two DNA strands will unwind. So these two strands will unwind. But they will not just stay like that and start to unwind. Something must be catalyzing that unwinding of the two strands. And the enzyme catalyzing that unwinding of the two strands is called what? Helicase. So this helicase enzyme will catalyze the breakdown of these hydrogen bonds, unwinding the two strands. So basically what we now have will be something like this. Will now be right? It's not my structure, but we unwind the two strands from each other. Unwind the two strands from each other, and here we are having, say, G, T, T, C. And here we are having C, A, G. All right. So the two strands now, they are not separate strands because you've broken the hydrogen bond that is holding them together. The next thing that will be happening after unwinding the two strands, each of these strands, remember one was template and the other one was complementary. But by the time you unwind the two of them from each other, both of them, the two strands become template strands. So this one becomes template for the synthesis of a new one. This one becomes template that directs the synthesis of a new strand also. So now, there is an enzyme called primase. Primase will direct the addition of a primer. Primer is small oligonucleotide that begins that transcription process. So primase will catalyze the binding of a primer here. So once primer binds, nucleotides will begin to add here according to the bases found here. So nucleotides will begin to bind to the primer, bind to the primer like that until we have this. So basically, look, as nucleotides binding, that is the ligase will help in doing that also. But now, look at this. How will the nucleotides know the right place to bind? DNA polymerase is there to proofread, to ensure that the right nucleotide binds to the right complementary strand, the right template. Now, if you look at this, we have an old template strand and a new one synthesized. That is why we say DNA replication is semi-conservative. Because for every new DNA synthesized, there's always an old strand and a new one attached to it. All right, so basically that's that. I'll put this into stages of DNA replication. This DNA synthesis is also called DNA replication. So if you ask to write on DNA replication, it's same thing as DNA synthesis. All right, now I'll talk about the stages, but you've been seeing this structure on the board since. There's what we call Okazaki fragment. Now look at this as your DNA. One strand which we can see as our template strand is the leading strand. Remember that the two strands will unwind from each other and one strand becomes serve as a template and will not direct the synthesis. Primer will bind, and as primer binds, will not be adding nucleotides there. That can be seen as a lagging strand. Now, addition of nucleotides to the leading strand is always continuous. So at all points, you'll be adding nucleotides in the leading strand. There's no gap. But on the lagging strand, which is seen as arising from the primer now, which will be serving as a complementary strand, there can be gaps in the addition of nucleotides. So this synthesis of new strand, this synthesis of nucleotide here is can be discontinuous. The addition of nucleotides here can be discontinuous, and that will form our Okazaki fragment. So our Okazaki fragments is actually short fragments. So the short fragments on the lagging strand of the DNA is called the Okazaki fragments. We have our ligase here that will join all those short strands together, filling all those gaps. So we have our normal DNA strand. So the Okazaki fragment is just short fragments on the lagging strand of the DNA. All right. So that being said, the stages of DNA synthesis can be summarized into initiation stage, elongation stage, and termination stage. Now, the initiation stage, this is our nucleus, and we have chromosomes in your nucleus. And on the chromosome, DNA is found on the chromosome. Now, the initiation phase is actually when that portion, one portion of the DNA there starts to unwind. And I've said before that the portion, that particular region where replication starts on the DNA is called what? Origin of replication. And this initiation is initiated by helicase. Helicase is the enzyme that will unwind that two strands from each other by breaking the hydrogen bond. And that unwinding starts from the origin of replication. So once the unwinding has started, that is the initiation phase. Then after the two strands unwind from each other, we said each of the strands will now serve as templates for the synthesis of a new strand. So nucleotides, primers will attach, nucleotides will start to be adding to the second strand. That is elongation. That's elongation. So once you are done with that, you are done adding the nucleotides. The nucleotides have been added, and we've replicated the whole chromosome or a good portion of the chromosome. Then termination will take place. So DNA synthesis will be brought to an end. We stop. There will be a stop. So there will be a stop signal to stop that replication process. So basically, we can summarize everything into initiation, elongation, and termination. Termination is the ending of it. So if you have a strand like this, let's say GAT, and all these things, the complements have been formed, then obviously it will be brought to an end. All right. So basically, this is a brief of the synthesis of DNA. So if you ask to write on the synthesis of DNA, please tell them that the first thing is that the two strands will unwind from each other, starting from the origin of replication. After the two strands unwind, each of the strands will serve as templates for the synthesis of new strands. Then a primer will bind. As primer bind, nucleotides will start to be added. So you explain all those for them. All right. Then you can tell them the function of all this. DNA polymerase will proofread the strands to ensure that the right nucleotide is added. That's why DNA polymerase is called a professor, because it's always reading the strand, always reading. Helicase will unwind. Primase will attach that primer. Ligase here, we join the short segments of the Okazaki strands, fragments together. All right, so basically, this is. If you have any question, please drop it in the comment section.